This is Biochemistry Unfolded, from atoms to algorithms, a weekly podcast on the topic of computational biochemistry and biophysics. Welcome. It's February 3rd, 2025. Welcome to the third episode of my biochemistry podcast on Biochemistry Unfolded, from atoms to algorithms. And so in this episode, I wanted to talk about something that's been kind of elusive. And if you've seen, I think the Indiana Jones episode with the Holy Grail, right? And so if you remember the Holy Grail, the cup of Christ, choose wisely. And this has also been kind of a synonym. The Holy Grail has been a synonym for problems that have been difficult to solve over the years, both in science or just overall. And so what is the Holy Grail of molecular biology or biochemistry? And I think that the Holy Grail has long been, what is the process of protein folding? How do proteins go from a single structure, a linear sequence, to their folded tertiary and quaternary structure? And so this has been ever since we've, begin to look at protein structure in the 20th century. This has been an elusive problem that has taken many years to solve. And we've made a lot of strides in determining how proteins fold, but we still have a long way to go even today. And so there's two main papers that go back in trying to answer these problems. And so the first paper came out in 2011, and that's a paper on how fast foldings fold by Kresten, Lindorff-Larsen, Stefano Piana, Ron Grohr, and David E. Shaw. So they're out of the D.E. Shaw Research Group in New York City. David E. Shaw, interestingly enough, David Shaw, he's a billionaire hedge fund manager, so he became very famous and fairly wealthy. He's a billionaire. He became wealthy through the hedge fund industry in finance, and he's also a molecular biochemist and a biophysicist, I should really say. And so he's been very interested in protein folding. And so he founded a company, D.E. Shaw Research, which is based in New York City. And they built a supercomputer for solving this particular problem called Anton. And so what is Anton? Anton is a highly specialized, massively parallel supercomputer. So when you're running in parallel, that means you're executing multiple strings of commands all at the same time. And so in 2008, D.E. Shaw Research in New York City built the supercomputer called Anton, which isn't a huge supercomputer by the standards of Frontier and some of the supercomputers at national laboratories with the Department of Energy, that they have a million or more processors and protein cores. The Anton machine, the original Anton machine, is only a 512 node supercomputer, but it's capable of performing, the original Anton was capable of performing 17,000 nanoseconds of simulated time per day for a simple protein water system. And that system would only consist of about 23,558 atoms. So not a very big supercomputer, but it was capable of performing multi-petaflop floating point operations per second simulations over time. Anton is so named because, well, it was named after Anton van Leeuwenhoek, the Dutch scientist who's literally invented the light microscope. So he's kind of considered the father of microscopy. And so now the Anton supercomputer is able to look at the simulation and dynamics of several proteins and at a very fast rate. We're now up to Anton 2, which is available, and the National Institutes of Health supports Anton, and they actually have an Anton. The Anton supercomputer is actually located at the Pittsburgh Supercomputing Center, which the University of Pittsburgh and Carnegie Mellon University in Pittsburgh work closely with that. I think technically PSC, the Supercomputing Center, is physically at Carnegie Mellon University, although literally those two schools are like right across the street from each other, right? So we do have to give credit to Carnegie Mellon University, of course. So Anton's pretty cool. Now, one fact about Anton, each Anton contains 2 computational subsystems. And so most of the calculation of electrostatic and van der Waals forces is performed by the high throughput interaction subsystem. That's the HTIS. And so this subsystem contains 32 deeply pipeline modules, and they're running at about 800 megahertz. That are arranged very kind of in an array, right? And so then the other calculations are done by the other subsystem, and that includes bond forces, and then they use fast Fourier transforms to do the long-range electrostatics. Usually that's done by a PME electrostatic system. And so then that subsystem contains 4 general-purpose tin silica cores and 8 specialized SIMD cores called geometry cores. And so that runs at 400 megahertz. So not terribly fast, but the way it's worked, it's got a 3D torus, and each chip has 6 interconnected node links with a total bandwidth of 607 gigabits per second. You say giga or giga? I don't know. I usually go by the popular movie pronunciation of giga, right? Doc Brown. But anyway, so the internode link is composed of 2 equal one-way links, and then you've got a one-way link having 50.6 gigabits per second. And so you've got a very fast supercomputer. You've got the one-way link has 11 different lanes, and each lane is a differential pair of wires at 4.6 gigabits per second. So the per hop latency is going to be about 50 nanoseconds. And so through the memory, each ASC has its own RAM bank, DRAM, and that enables extremely large systems right within the node itself without having to go towards to explore all the latency of everything else. So you can run very fast, extremely fast simulations right there on Anton. What's also unique about Anton is it's capable of performing a simulation, and the hardware has been designed by the ground up. So, for example, an Anton supercomputer doesn't necessarily have Intel inside. The CPU, the GPUs, all of the hardware was designed from the ground up at D.E. Shaw Research and to run molecular dynamics simulations. So it was designed specifically to run molecular dynamics. There's actually no real operating system, so you don't have that overhead of running an operating system. The supercomputer has as many CPU cycles available to run its molecular dynamics That's another reason we can get an extremely fast simulation. And the current versions of Anton, you can actually, academic researchers are available to utilize Anton for their own research, and so you can actually apply for time through the Pittsburgh Supercomputing Center and DE Shaw Research if you have specific things you're interested in. So it's a very useful supercomputing center, or a very useful supercomputer. Now the fast folding protein paper, this is one of the first papers, and again it came out in 2011, and it investigates the fundamental principles of protein folding using long-range timescale molecular dynamics. And so there's 12 structurally diverse proteins they looked at, things like chignolin, tripcage, which has tryptophan in it. That's a very popular protein. BBA, Villain, WW domain, NTL9, BBL, protein B, homeodomain, protein G, alpha 3D, and lambda repressor. Now what's unique about these proteins are all they're called fast folding proteins. And so what that means is these are small proteins, no more than about 100 amino acids or less. In many cases, it's usually less than about 60 amino acid residues. They're all a single chain, and so they're simple. They fold without any post-translational modifications. They don't require a ligand to be bound. They don't require a secondary protein to interact with. So there's no quaternary levels of protein structure. And of course, all of their structures are known and available in the protein data bank. So we're able to fold these 12 proteins at atomic resolution. What that means is we've got an all-atom molecular dynamics simulation. And for this, for molecular dynamics, now they're using molecular mechanics applied with Newton's laws of motion, and so we're calculating the force and the acceleration on the atoms to give us an actual simulation. For that, they used the CHARM force field. CHARM was developed out of, interestingly enough, Martin Karplus's lab. He's known for CHARM. Now they did use a modified CHARM force field. So they had to kind of modify a couple things with CHARM in order to get that to work, right? So for one, they had to modify the backbone torsional parameters to ensure that there's a better agreement with experimental structures and quantum mechanical calculations, and they had to modify some of the side chain dihedral parameters as well, to correct deviations from previous CHARM versions. They've also had to look at some of the solvent-protein interactions to balance hydrophobic and hydrophilic interactions. And so this is important because proteins exist in an aqueous medium, but at the core, what we know about protein folding is that when a protein folds, it undergoes hydrophobic collapse. So the hydrophobic amino acids organize mainly towards the middle, with the polar amino acids on the outside. And so in order to model this, we had to tune the force field to optimize the balance between these hydrophobic and hydrophilic interactions, right? The force field was also modified for refinements in hydrogen bonding and secondary structure stability, because when a protein folds, you're going to form secondary structural elements. You start to form alpha helices and beta sheets. These are stabilized by hydrogen bonding interactions. And some force fields tend to overstabilize or destabilize these secondary structures. And so you have to make some adjustments to those hydrogen bonds to ensure that you have stability of the alpha helix and the beta sheet. So then, you want to test against multiple classes, so you don't want only alpha helices or only beta sheets, so you need some proteins with a little bit of both. And so several of these proteins, like protein G and NTL9, have both alpha helices and beta sheets present. I think there is still a little bit of bias in these calculations because many of these are dominant more towards alpha helices than beta sheets, but that is one of the modifications that they looked at here. So they used a modified charm force field to enhance specific interactions there. Now, so with the simulations they did, and these are all very long simulations, if I kind of look at some of these, we're looking at anywhere from 0.6 microseconds. And so a microsecond is like 1,000 nanoseconds. When I was in graduate school, I think I was lucky enough to get maybe a picosecond, a couple of picoseconds of simulation time. But now we're on the nanosecond, 1,000. 0.6 microseconds is 600 nanoseconds. Some of these simulations took 18 microseconds or 29 microseconds. I think where's the longest one on here? I think we have one. Protein G was 65 microsecond simulation. That was a pretty long simulation. And so when you simulate, when you simulate a very short amount of time, there's not much dynamics you can do. The longer that you simulate, you can actually start to look at major domain movements and fluctuations. And so when you simulate for 65 microseconds, then you can compare the RMSD to the crystal structure, and that's the root mean squared deviation. And then you can really look at how that protein folds overall. And so what they found is that proteins fold spontaneously and repeatedly to their native structures. So you would kind of undergo a spontaneous folding, you'd find the native structure, and then it would kind of unfold a little bit and then fold back. And it also generally follows a single dominant route with the order of structural formation that correlates to the unfolded state's propensities. And proteins would also exhibit multiple folding pathways. So it's not just one, it's also a very deterministic process. So you kind of follow a path, kind of like you're going through the Smoky Mountain National Park with the different valleys that would represent low energy, and you're following a very specific path to that folded protein. So it was very deterministic. Early in the folding process, the proteins would acquire a native-like topology with partial secondary structures. This kind of follows the formation of what we call molten globules, and then the tertiary context would form as the molten globules came together. So we kind of observed that formation right there. So we know a lot more about the processes and the dynamics of protein folding from this paper right here. And so some of the key results, 11 out of the 12 proteins folded to within a 2 angstrom root mean square deviation of their crystal structure, their experimentally determined structure. And so that was actually fairly significant. I think there was only one protein that folded. I think it was possibly protein B that folded to more than more than 2 angstroms. Now, generally, when you compare an RMSD, if the RMSD, the difference is less than 3 angstroms, you've got a very high quality structure, and so that's considered kind of the gold standard. So with 2 angstroms, that's considered very good, right? And again, the simulations also provide estimates of various free energy barriers, With subproteins like BBL and protein B, they showed a downhill folding without a significant energy barrier. So that they looked at those energetics as well as the kinetics. And so they found that, for the most part, we have very high resolution structures, and it confirmed to follow the experimental data. Now, what could this mean for science? Obviously, we've learned a lot about protein folding, and we can make, since 2011 when this came out, we've really looked at molecular modeling, and molecular mechanics force fields have increased. It was only 2 years later that Martin Karplus actually won the Nobel Prize for his work partially on the charm force field, right? And so this also highlights the reliability of using molecular mechanics as well as molecular dynamics to study protein folding. So that was established. That was a good thing, right? This could also lead to further developments in understanding protein misfolding. Now, when proteins misfold, such as in the amyloid plaque of Alzheimer's disease, Parkinson's disease also involves misfolding. So if we can study if we can understand how proteins fold, we can also have a better understanding of how proteins misfold when that happens, when there are errors that take place. And this will help us understand diseases like Alzheimer's and Parkinson's disease in order to hopefully find a treatment or a cure for those diseases. Furthermore, of course, with protein design and engineering, the insights from these simulations would improve de novo protein design. This kind of leads us to the recent Nobel Prize in Chemistry last year in 2024, where especially David Baker's group has developed Rosetta for understanding how to not only, you can basically take the structure of a protein you want to try and do something new with and design that protein from scratch, right? It also helps us understand things like AlphaFold 2 and be able to predict a protein structure from its primary sequence. So this paper kind of led, in 2011, this was kind of a stepping stone towards the development of AlphaFold and Rosetta as well. So it's very important. It also kind of provides, the paper also provides the key selling point towards the Anton supercomputer, where we can use a highly specialized piece of hardware, Anton, and now Anton 2 is the new one. And so that further develops the use of that for molecular simulations. And of course, we've made several steps in the refinement of the force fields used in these simulations as well. So this was a very important paper, and I think I think if you look at Google Scholar, let me just kind of see Google Scholar here. Let's see how many. I want to just see how many. How fast folding proteins fold. And so if we look on Google Scholar, this paper has now been cited by 2,160 other papers, right? And so if we just look in the last year, it's actually still being cited. Here's a paper on enhanced sampling with machine learning. So again, this is setting, actually there's several papers in here with AI. Here's another paper, ab initio characterization of protein molecular dynamics with AI BMD. That's also, the paper's been cited by that paper. We have a citation in a paper on using structure prediction using quantum computers. And so quantum computers are a whole new field which should greatly increase the speed. So again, Anton 2, kind of its own specialized hardware. Now we can go and, if we could use quantum computers and develop that, that's where things are going to go. And there's over 2,000 papers that cite the Lindorff-Larsen D.E. Shaw paper in the research. So that's pretty good. Now the other major paper that kind of sets that kind of helps us find the holy grail of biochemistry and molecular biology, right? Because that's ultimately what we want. We want to choose wisely, right? So the other major paper, of course, came out in 2021. This was about only 3 years before AlphaFold 2 won the Nobel Prize just this past year. And so AlphaFold was largely introduced by this paper in Nature, right? And so the paper in Nature was written by the authors. Now there's a lot of authors on this. John Jumper, who is one of the 2024 Nobel Prize recipients. And Demis Hassabis is the last author on this paper. He also received the Nobel Prize. Now there's a whole bunch of other scientists on this paper: Richard Evans, Alexander Pritzel, Tim Green. I'm not going to list all these names, but you can kind of, if you look at all the names, you can see 2 of them won the Nobel Prize, and there's a lot of, that kind of highlights the importance. When we look at a Nobel Prize, it's awarded to 1 or 2 people. For every person that receives a Nobel Prize, there's probably 20 or 30 that contributed to that research. So science is not done by just one person anymore. Science is done by a large team. So this paper is Highly Accurate Protein Structural Prediction with AlphaFold, published in 2021. So I think we can kind of say this certainly is, this introduced what AlphaFold does. And so obviously we're taking as input the protein's primary structure, and we're predicting as output the 3D structure, right? And so this uses artificial intelligence. We're using AI. And so AlphaFold 2, this is a deep learning based computational model that's developed by DeepMind, which is a subsidiary of Google. And that we predict the 3D structure of proteins, again, with atomic accuracy. So what that means, just like in the D.E. Shaw paper where they had atom level accuracy in molecular dynamics simulations, we're now predicting the atomic coordinates of a protein in its 3D structure. And so we integrate AlphaFold integrates multiple machine learning techniques, and this incorporates both evolutionary as well as physical constraints of those proteins. And so that's so some of the key aspects of AlphaFold. There's 3 main key aspects of AlphaFold. Number 1 is its neural network architecture. And so again, a neural network is kind of a computer doing kind of what the human brain does, right? And so the way neurons are connected in the brain, you've got one neuron that's connected, interconnected to multiple other neurons, right? And each neuron depends on all of its interactions with everything else to come up with, to solve problems. And so a neural network is built on that same basic framework. And we call this a deep learning framework because the computer can actually go through multiple iterations and learn as it goes. It gets substantially better with each iteration that it puts through. And so this novel deep learning framework combines multiple sequence alignments. We're taking the sequence of the protein, we're then looking at a blast search to find similar sequences, and then we're looking at pairwise residue interactions in that multi-sequence alignment based on everything that's available of that structure, right? At the core of AlphaFold is something called EvoFormer. And so what is EvoFormer? This is a neural network block that refines the MSA information, and it extracts all of the structural constraints. So the EvoFormer is looking at how all those residues interact with all the other residues in the known data to be able to then make predictions of what residues it would expect to interact with. And so there's a structural module which then directly predicts the atomic coordinates iteratively using geometric transformations. Number two, the second major aspect is the end-to-end prediction. And so we're starting from the amino acid sequence, we're then searching for evolutionary homologs, and then we're predicting the interaction, the interresidue distances and orientations. So we're refining predictions iteratively and leveraging recycling through a feedback loop to improve the accuracy. Number three is its performance. Now, back in 2021, there's a system called the Critical Assessment of Structural Prediction, CASP for short. And so I think this was CASP 14 that was done. And so this particular paper highlights AlphaFold's use or its domination, if you will. It literally killed all the competition in CASP 14. So it's a competition that they basically give you a protein, and if you're developing protein folding software, you're supposed to be able to predict that structure. Well, AlphaFold predicted, it blew away everyone else, literally, and it predicted with a median backbone accuracy of 0.96 angstroms of root-mean square deviation against everything else in the CASP data set. So 0.96, anything less than 3 is very good. We're less than 1. And so AlphaFold had a huge prediction. So that, that certainly, and 3 years later, Nobel Prize. So that's where we kind of are. We're pretty much, we don't quite have the Holy Grail. There's still a lot of work to be done, but we're pretty darn close, right? Yeah, so that's pretty cool. So we're dramatically improving protein structure prediction, and then we're learning from that sequence data, and we're predicting the predictions can be compared to high-resolution X-ray crystallography or cryo-electron microscopy structures, which are 2 experimental structures that are kind of the gold standard, if you will, in biological structure prediction, or determination, I should say. Now, where is this going to go? The future. We can look at enabling predictions of the proteins of the whole proteome. Now, if you've heard of genomics, the genome is, all of the DNA, all of the genes in an organism. The proteome is all of the proteins in an organism. And if we can predict the structures of all those proteins, can you just imagine what that's going to do for developing drugs of tomorrow, or even designing new proteins, protein engineering? And even in synthetic biology, designing entirely new biological structures. So this is going to greatly accelerate, AlphaFold is going to accelerate that information. It's also going to help us understand the biological significance of protein folding and function and interactions in a wide variety of proteins. Biophysically, we can apply this to protein-protein interactions and ligand binding. Now, AlphaFold 2, that's AlphaFold 2, doesn't really work with protein-protein interactions and ligand binding, so that's kind of the weakness. We already have AlphaFold 3, and so that just came out in the past year. It initially came out as a closed source program. People didn't like that, but I think one of the pharma companies had some intellectual property that they wanted to use that for drug discovery, and so it was closed source. The scientific community likes things to be open, and so we can actually look at how things work, not necessarily so we can repeat it, but we want to verify those results. Verification of results is part of the scientific method, right? So AlphaFold 3 can look at things like ligand binding, protein interactions. We can also look at things not just with proteins, but also RNA and DNA structures. So that's kind of where all this is going. We're going to be able to essentially simulate. We're rapidly achieving the capability of simulating everything that takes place in a human cell on an atom level, if you can believe that. So we can put all that into a computer and look at all of those interactions and how it takes place. So some of the limitations, of course, I've kind of hit on one of the main limitations with protein-protein ligand interactions and ligand binding. So that's one of the main limitations. AlphaFold 3 is helping to address that. Another limitation is post-translational modifications. So where, when proteins fold, they may be modified in the endoplasmic reticulum by post-translational modifications after the fact. And how does that implicate into things, right? And things like cofactors as well. The other key limitation is the hardware. Yes, you can download AlphaFold 2 source code from GitHub, and you can run it. You can run it on Google's hardware. You can download all the databases you need on your own computer as well if you have that, but you need a computer with a significant amount of capabilities. You need GPUs. You need a lot of hard drive space for all of the databases that you need. So that's why it's mainly run on supercomputers today. So it's still, and GPUs, you need state-of-the-art GPUs because it's fairly memory intensive. Of course, these limitations apply to anything AI. So you need a massive supercomputer, such as the one Elon Musk is building in Western Tennessee right now. That's a massive one, or any of the other supercomputers that are available and optimized for AI calculations. So that's a big thing. One of the other key limitations and concerns is bias in the model, because we already have a pretty good structured database, and if you're predicting a protein whose structure already exists in the database, or a protein who's got where you've got a very similar structure, how does AlphaFold not just take that structure and say, hey, here's the structure, I just found it, right? So you're not really predicting anything. But AlphaFold doesn't actually do that, right? It's not a method of retrieval. AlphaFold's methods are a method of actually folding that protein, right? So it's doing end-to-end structure prediction, starting with the data in the multi-sequence alignment and homologous sequences, and it's predicting interresidue context from that, as well as the angles to fold from scratch, using its own deep learning technique. So it's not actually retrieving the structure, even though, yeah, you can, it finds this, it looks the sequence up, it can find a sequence and maybe fold it from there. But some people are some critics are like, hey, it can just find the sequence. Okay, let's just find it. Let's do that, right? And so we want to handle known structures as templates, and then it can use that template as a reference, but it doesn't use it as actually direct output. So it only incorporates broad structural constraints from templates, and then it recalculates the entire protein structure using its own algorithms for determining how things are connected, right? Now, when templates are low quality or missing, then AlphaFold 2 will rely on the multi-sequence alignment and deep learning for an accurate prediction method, right? When it comes to structural bias, it kind of prevents memorization of training data so that the model is not explicitly trained to recall specific structures, but rather to learn generalizable folding principles. And so when we present, when it's presented with a sequence in the PDB, it's going to still run its full prediction pipeline rather than copying stored coordinates. So it still uses its own algorithms. If you look at the source code, it's still actually doing all of its own prediction, right? And so AlphaFold 2's prediction may slightly differ from the experimentally determined structure. And we can actually see evidence of this because we can get a pretty good backbone prediction, but you might have two side chains, one's pointing in one direction, another side chain's pointing in the opposite direction. We've also seen this in homology modeling when predicting protein structure with a homology model from a template. Essentially, AlphaFold 2 is doing something very similar to homology modeling. So again, you're predicting structure, and you know, when they did CASP 14, right? They went with previously unpublished structures that were unknown to the model, and so it predicted that completely from scratch. So there was nothing that it could really look up there. It predicted all those from scratch, and then later on we can align those structures and say, hey, this actually matches the experimental structure. But those experimental structures were not available at the time AlphaFold was tested in CASP 14. So that kind of helped confirm that it was actually predicting those structures and not just finding something that was available in the database, right? And so for completely novel proteins, AlphaFold 2 can make accurate predictions using only sequenced data. And it's particularly effective when deep multi-sequence alignments exist, even if we don't have any homologous structure that's known. So that the model can successfully predict structures that are experimentally validated. So AlphaFold 2 doesn't just find and reuse known structures. It applies those deep learning methods to predict those structures, what we call de novo, or from scratch, using its own structure prediction methods. And so it's able to obtain that data, right? So hopefully that kind of helps us deal with some of the bias in the structures, right? So that kind of helps us predict the Holy Grail. That kind of solves the Holy Grail. Of course, we still have a lot of aspects to go. That the AlphaFold 2 is going to revolutionize, and it's going to continue to revolutionize the field of protein folding and protein structure prediction. I think in the next years, if you think about it from machine learning, AlphaFold 2 is like an infant. It's less than 5 years old. Can you imagine where we're going to be when AlphaFold is 20 years old or 30 years old? it's going to be, yeah, in those years, we have some pretty exciting times ahead. So it's going to be pretty exciting. So that kind of concludes our episode of Biochemistry Unfolded, from atoms to algorithms. I hope you found this very interesting. And so if you like this episode, go ahead and subscribe to our channel here, and when we post new episodes, you'll find that. You can find us on Facebook and Instagram as well too. And if you like this, go ahead and click the like button, and you can, if you have any questions, leave a comment. And if you have anything you're interested in the field of biochemistry instruction, let me know, and maybe that will be covered in a future episode. For now, thank you, and I will see you again. Thank you for watching Biochemistry Unfolded, from atoms to algorithms. If you enjoyed this content, please subscribe to this channel, and we will keep you informed of future episodes. Thank you.